So hello, everyone. I'm Max. I'm a graduate student in the post-cancer lab at UCSF. And today I'm going to talk about analyzing astrocyte calcium data at the network level. And in particular, going to focus on how viewing astrocyte calcium data as discrete calcium events facilitates analysis by enabling the compression of data while still maintaining important information about the dynamics of astrocyte behavior. So just to motivate the discussion a little bit, I think this has been reiterated many times, but astrocytes form an interconnected network of highly ramified cells that extend processes out to tens of thousands of synapses per individual astrocyte. And this sort of real estate that astrocytes occupy inside of the nervous system really gives them a prime position to be able to regulate the activity of large populations of neurons. And we can measure this activity of large populations of neurons readily using techniques such as population electrophysiology, like the local field potential. So when large groups of neurons change their aggregate dynamic behavior, we have changes in sort of the oscillatory dynamics that we can measure using the LFP, for example, from lower frequency to higher frequency oscillatory behavior. While astrocytes don't possess the same electrical characteristics that neurons do, astrocytes carry an incredibly rich repertoire of calcium dynamics. And calcium plays an incredibly important role in the behavior of astrocytes because of its central location in many different pathways. And so here is an example of a calcium imaging data set taken from Cortex measuring astrocytic GCAMP in vivo. And calcium imaging is incredibly important because it enables us to capture so much of the rich detail in the dynamics in the spatial and temporal behavior of the calcium fluctuations within an astrocyte. However, it carries a tremendous technical challenge in that because the data is space by potentially space by time, the high dimensionality of these signals poses a tremendous challenge for analysis. So as was beautifully presented yesterday by Dr. Botiang Yu, the tool Aqua, which has been recently developed, facilitates analysis of this high dimensional astrocyte calcium imaging data set by separating out the raw signal into individual discrete calcium events, which then have their own quantified features that are much more conducive to analysis. And what this enables is the ability to view the astrocyte data through different lenses that compress various features of the large high-dimensional raw imaging data set into these views that maintain important information content about the underlying dynamics. So here in this video, we see the same video from the previous slide, but now superimposed on the right, we see the Aqua segmented events with the colored overlays. So an individual Aqua event contains a lot of information, but it can be distilled down into various facets. So, for example, an Aqua event might contain, or an Aqua event does contain the spatiotemporal profile of the activity of that particular event's fluorescence dynamics. But you can also sort of consider the event as being flattened in space to really only consider the time evolution of the fluorescence dynamics in that particular event. So in that way, you sort of compress away the spatial component of it to only look at the time trace. If you wanted to, you could even just get rid of the dynamical component of it completely and only consider it to be a discrete point in time also, and only look at the onset time. So we'll talk about how that sort of perspective can also be helpful for facilitating analysis here. But one of the critical components of Aqua is the generation of these feature vectors associated with each individual event that quantify various aspects of the event, including its spatial characteristics, such as its area or circularity, these various quantifications about this shape, the temporal sort of dynamics of the event and its kinetics, duration, rise, fall times, the sort of synchronization of many events within the same imaging field and how they overlap with each other in space and in time, as well as the propagation of events in space, because some events are static, whereas others will actually change in their position relative to the imaging field over time. So all of these events get quantified and packaged together into feature vectors that are associated with each individual discrete event. So we're really interested in the regulation of network dynamics by astrocytes, and the sleep-wake cycle provides an ideal in vivo model of this, because the sleep-wake cycle induces large changes in the network dynamics, which we can readily measure with population electrophysiology. So here today, I'm going to be focusing on a study that was recently published by Trisha Vaidyanathan in the lab, looking at at astrocyte behavior during the sleep-wake cycle. And even you can see in this LFP trace just showing filtered signal that there's a large change in the oscillatory dynamics in this particular frequency band in sleep periods in red versus wakefulness. So in this particular setup, we recorded simultaneous 2-photon astrocyte calcium imaging with 2 electrophysiological measurements, a LFP electrode that was placed proximal to the imaging window, as well as an EEG electrode that was placed far away from the imaging window. And then, as I mentioned previously, the aqua was used in order to identify discrete calcium events from the 2-photon imaging data. So I'm going to focus on 3 different analyses that can be used based on the events that are generated by aqua. And I'm going to sort of talk about 3 different insights that we can gain on the basis of different views of those events. So first, I want to talk about how events are interrelated with one another. So as I mentioned before, the astrocytes form this highly interconnected network. There's known extensive anatomical connectivity between astrocytes that form this beautiful syncytium. So if you took 2 spatial regions, for example, the orange and the purple regions that I've highlighted here, and you asked a question of, well, how are those 2 physically connected? You can draw a line, in space that forms a cable, if you will, or a physical connection between those 2. So there's anatomical connectivity. However, the functional connectivity or the information flow between different regions of the astrocyte network is less understood than the anatomical connectivity itself. And so in this particular analysis, I'm going to be focusing on using the spatiotemporal coordinates of individual astrocyte events in order to gain information. So in order to identify connectivity between regions in the network, we're going to use the spatial location of an individual astrocyte event. And in order to identify reliable timing relationships, we'll use the temporal coordinate of the event. So as I said, the general strategy here is that although we know that there is a physical link that goes between 2 regions of the network that is traversable, we actually have more information when we have this recording of the calcium imaging that we can identify events from, because we can look between 2 different given regions of interest within the network and identify reliable timing relationships between the events that occur in those 2 regions, and use that to infer that there's information flow between those 2 regions of space. So the metric that we used in this particular case to infer this connectivity was transfer entropy. And transfer entropy is based on the principle that although there's a certain amount of intrinsic uncertainty in the behavior of one signal in one region of interest based on its own history, the uncertainty can actually be reduced if we have knowledge of a second region of interest behavior in the past, as long as if there is some type of reliable timing relationship between those 2 signals. And actually the degree to which that uncertainty is reduced, so the amount of the change in the uncertainty that we have by gaining the knowledge of the second region of interest signal in the past, actually gives us a quantification of the degree to which this information allows us to improve the prediction of the original signal in the present. And this quantification is precisely the transfer entropy. It's the improvement in our prediction or the reduction in our uncertainty, thought as 2 sides of the same coin, that we get by knowing a second signal. So the region-to-region functional connectivity is a bit difficult to visualize because it's 2-dimensional by 2-dimensional, so it requires 4 dimensions of space to sort of visualize. But for as a shorthand here, what I've done is I've taken the imaging field that we had in this particular setup and divided it into these 16 regions of interest in either direction. And so here, what I'm highlighting is for the region of interest in magenta here. This inset plot is showing the normalized transfer entropy from the magenta region to the rest of the imaging field. So we can see its projections of information flow to the rest of the imaging field. And if we look at this across the entire set of the regions of interest that we examine, although it's a little bit difficult to visualize, by focusing in on a couple different sections that I've shown, so the blue region of interest and the red region of interest, what's really striking is that the patterns of functional connectivity that each of these astrocytes have, or each of these regions of interest, have to the rest of the imaging field, are actually quite distinct depending on spatial location. So there is spatial heterogeneity in the patterns of functional connectivity between astrocytes. And if we apply some graph theoretic measures to this by measuring network degree for individual ROIs, we can identify sinks, which have large numbers of incoming weights, and sources, which have large numbers of outgoing weights in the network. And so if we apply this analysis and look across all the regions of interest within the imaging field, what we see is that there actually is, there are regions that are spatially distinct that serve as input hubs, or areas that have a large input degree, or output hubs, or areas that have large output degree. And these areas are actually spatially distinct. So by viewing aqua events as coordinates in space and time, we were able to see that astrocyte-astrocyte functional connectivity or information flow is spatially heterogeneous, and that there are distinct input hubs or sinks and output hubs or sources. So next I want to focus on the feature vectors that are associated with individual events and how they change depending on context. So as Guoqiang Yu yesterday pointed out, and as we can see here in this video, the astrocyte calcium events are highly heterogeneous. So here, once again, we see a calcium recording from ex vivo cortical astrocytes on the left side, and on the right side, we see an overlay of the astrocyte events that have been detected by aqua. And we can see that the characteristics of their fluorescence are quite diverse. And aqua captured this beautifully by providing with each individual identified event a vector of features that sort of describe quantitatively how the event looks to a certain degree in terms of its spatial characteristics, its dynamics, its propagation, and so on. So a natural question that are all of the features that an astrocyte event might be able to occupy, right? And building off of that, whether there are actually changes in this space depending on the context, for example, the network state of sleep versus wake, or based on the GPCR signaling milieu of the astrocyte. So by using PCA on these identified feature vectors, we can identify that there are covariating features of astrocyte events that actually explain the majority of the variance, the very small number of principal components. So there are axes inside of this feature space that explain the variance in the behavior of the individual event features. And they break down very nicely in terms of features that are related to sort of the spatial extent and propagation of the event, a principal component that's related to sort of the temporal dynamics, the duration and rise and fall times, and then a third principal component having to do with the df over f amplitude of the entire event, as well as temporal overlap between them. One of the principal findings of the work by Tricia in this report was that by activating GI dreads, we were able to manipulate the GPCR signaling of astrocytes by specifically activating GI dreads, and that this caused an increase in the calcium event rate that we saw over time. So in the cumulative distribution over time of astrocyte events for the active CNO condition versus the saline controls, we saw just an overall increase just quantitatively in the number of events that were observed in imaging. But that wasn't the only change that we saw in the GI dread active condition, because what we actually found was that the activating the dreads resulted in changes in the characteristics that occurred. So if you look in these particular feature axes where these events are in the feature space, we actually saw that activating the dread resulted in changes in particular PCs, as we see here in the cumulative distributions. And what's more, we saw that the GI dread effect, which we measure as the Kolmogorov-Smirnov distance between these two cumulative distributions for the control versus the active CNO condition, actually depends on the cortical network state. So the effect of the GI dreads actually is dependent on whether the animal was asleep or awake. So for example, for PC2 here, the effect of GI dreads on PC2 was larger in the sleeping condition than it was in the wakefulness condition. And we can go back using the PC weights and actually identify, well, what were the characteristics that were part of the characteristics of these events that were differentially regulated by GI-GPCR signaling in these two conditions. And we can see that for PC2, for example, as I mentioned before, this is related to duration, rise and fall times. And for PC4, this is related to sort of the amplitude and the spatial overlap or synchronization of these events. So in this particular case, by viewing events as feature vectors, we were able to identify that there is a low dimensional sort of representation in which the event features covary, that activating GI dreads alters the characteristics of event features, and that this effect of GI dreads actually depends on the cortical network state. So there are components for which there is a larger effect of the GI dread manipulation in the sleeping condition than in the wakefulness condition. So finally, I wanted to take the opportunity to talk about how these astrocyte events relate to simultaneously recorded neuronal signals, in this case, LFP and EEG. And again, what we found was that astrocytic GI dreads actually induces changes in slow wave activity during sleep, both in the local LFP electrode that's proximal to where the imaging window and the GI dreads, the activation of the GI dreads were, as well as in distant sites that were recorded in the contralateral frontal cortex, so on the opposite side. So both locally and in distant regions, GI dread activation in astrocytes causes changes in slow wave activity during sleep. So a question that naturally arises then is whether local astrocyte events actually alter interactions between local and distant regions of the brain. So the measure that we used to identify this was coherence, which is the consistency in the phase relationship between the two signals at a particular frequency. And coherence between brain regions has been hypothesized to index or perhaps facilitate the flexible routing of information between brain regions by aligning the active periods, the more active periods of membrane potential in order to create a window for synchronizing when action potentials are able to propagate from one region to another. So in order to investigate this, what we did was we looked at windows around astrocyte events and windows around randomly selected events that had nothing to do with astrocytes. And we computed coherence between the two signals, the local and the more distant signal, in windows that were chosen on the basis of those two alignments. So in this particular case, our view of the events is just the onset time of the event itself. So we've compressed down all of the information of the fluorescence dynamics of the astrocyte behavior down to a single time point representing the onset of the event. And in doing this, what we found was that in astrocyte locked periods, there was actually increased coherence between the local and the distant signal that we recorded in a particular frequency band between 5 and 10 hertz. And that was increased in the coherence between these two regions in astrocyte locked specific windows, but not in randomly chosen windows. And what was particularly interesting about this was that this effect of astrocyte locking on the coherence was not uniform across the sleep-wake cycle, but actually specifically if we were looking at sleep, while the astrocyte locking effect here in green compared with the random locking effect in gray, as they seem to be overlapping each other for at baseline, in the 15 seconds leading into a sleep to wake transition, we actually see an increase in this astrocyte locking effect on the coherence between these two regions leading into this sleep-wake transition. So there seems to be, again, an effect of the cortical network state that on this astrocyte locking phenomenon. So to summarize again, viewing the aqua events as points in time, we were able to identify that interregion coherence is increased in windows following astrocyte events, and that this astrocyte event locking effect of coherence increases in the lead up from a sleep into a wake transition. So that was just a summary of a few different ways of viewing astrocyte events and how they can be used to sort of gain insights into the behavior of astrocyte calcium dynamics and how it relates to co-recorded neuronal signals. And to conclude, I just wanted to say that aqua, this fantastic tool, facilitates the analysis of high-dimensional calcium imaging data by extracting these richly informative calcium events. And that these events allow lenses, if you will, into the data that allow us to view different components of the dynamics, which makes possible a wide array of analyses and facilitates these analyses by compressing the high dimensional calcium data while maintaining that rich information content about the astrocyte dynamics. And with that, I want to thank the PostCancer Lab and the U Lab and plug Aqua again, which is available on GitHub. Thank you very much for your time.
Thank you very much, Max. Okay, looks like we have a question from Renaud. Let's see if I can unmute you.
Yeah, can you hear me? Okay, really cool stuff. I just had a quick question about your use of transfer entropy. Do you use a specific package for that? And did you do any kind of controls? I mean, can you give us a bit more details how you applied it?
Yeah, so in this particular case, I used the, I believe the package is called Smite. It's a transfer entropy package in Python that's available on GitHub. And what was really nice in this particular case that facilitated the analysis was that these events give a very nice it's already sort of turned into a binary raster essentially as a result of the event segmentation that's done by Aqua. And the Aqua segmentation, as was talked about yesterday in the presentation, really takes into account a lot of this this spatial information and that gives like structure to these individual calcium events, which really boosts your signal to noise ratio and facilitates these types of like information theoretic analyses. Yeah, we can talk offline about like particular ways of like the controls and stuff for the validation as far as the information flow for this. But I think the coolest thing about the event-based analysis in particular that really facilitates like doing these types of things is that by segmenting out these events that give you that allow you to take into account the information that's happening in nearby pixels and this kind of like smoothness constraint that you have on the spatial distribution of the data, it really helps to boost your signal to noise ratio in such a way that you actually can like, you have the information content to be able to like compute these types of things.
Okay, thanks a lot.
Thank you. And looks like we have another question from Carlos. I'm going to unmute you.
Hello, thanks for this cool work. My question is about the PCI applied to the different features, especially to the spatial features, because Aqua offers you metrics about the propagation according to the field of view. But of course, your field of view can change from one acquisition to another acquisition. Do you correct this, or you use like what kind of, how you correct, how you deal with this divergence, possible divergence between?
Yeah, so in this particular case, the propagation features that we were using were sort of directional, as you were saying. So they were related to like the cardinal axes, basically, of the imaging field. And so in this particular case, we used recordings that had the cardinal axes such that they were aligned to one another, so that like anterior propagation in one was like the same for all of the data sets that went into the analysis. So that it was a comparable feature between all of them. But Aqua has the ability to compute propagation in a lot of like other contexts too, not just like based on the cardinal directions of like the rectilinear layout that you have in like a particular recording. You can also do like, for example, if you have a landmark that you identified that you want to look at propagation in that particular feature, then you can look at like propagation toward or away from that particular landmark too. So in this case, we just took kind of a more simplistic approach for looking at the propagation as just being relative to like the, rostral, caudal, medial, lateral axes.
And don't you think that this can be like a sort of source of noise, or you think that astrocytes are like a polarized in some way according to these coordinates?
I think that's a really interesting question, honestly, as far as like what is the, is there anisotropy to the propagation features that we might see coming from, this type of analysis? I think that there's evidence. I think that there is evidence that in certain areas, depending on where you're looking at, that there are heterogeneities that are directional depending on like the particular, what direction you're looking at the imaging field from, that may possibly generate, some type of anisotropy in the propagation. I don't know if I definitively would say that like an up or down vote on that, but I think it's a hugely interesting thing to look at.
Yeah. Okay, thanks very much.
Okay, perfect. Thank you very much. And yeah, looks like we're good on timing. So we're going to start our next speaker, who is Audrey Denizot, and she's from the Okinawa Institute of Science and Technology in Japan. And her talk is on disentangling astrocytic calcium activity, insights from spatially extended models.
Thank you very much, Marcia. Can you confirm that you can see my slide and that you can hear me well?
Yes.
Perfect. Then, well, thank you very much. I'm very glad to be able to be here to present my work to you today. So I'm Audrey Denizot, and I am a postdoctoral scholar in Eric de Schuter's team in Okinawa. And I'm going to talk to you a little bit about astrocyte calcium activity, about which we discussed quite a lot already during this workshop, but trying to take a bit of a different perspective and showing to you what insights we can get from spatially extended models. So I will be very quick in the introduction because we had already amazing talks showing notably all of the roles that astrocytes can play in the brain, and here we're going to focus on their role in information processing. So here there is a video in which you can see calcium activity in a whole astrocyte territory from Agarwal and collaborators. And this video shows calcium activity recorded in vivo, and on the right side you can see the detected events. And you will see soon that the mouse will start running, and when the mouse starts running, there's a huge increase in calcium activity, as you can see here, which shows that this calcium activity in astrocytes seems to be linked with higher brain functions. And at the level of the synapse, those calcium signals seem crucial as well, notably for the interactions between astrocytes and neurons. So as most of you probably know already, astrocytes have ramifications that can be in tight contact with synapses, as you can see here on this electron microscopy image. And those are called perisynaptic astrocytic processes, and there they interact with neurons. So basically those processes express receptors for the neurotransmitters released by the active presynaptic neuron, and this triggers calcium signals locally. That can trigger the release of gliotransmitters that will then modulate neural communication. And thanks to the recent development of tools with higher spatiotemporal resolution, researchers in the field have been able to show more and more diverse calcium signals in astrocytes. So at the beginning, we thought that they were mostly slow and large events, but those were signals measured in the soma and major branches. And now that we can actually measure signals in smaller branchlets, there's evidence that most of this calcium activity actually occurs in very fine processes. And you can see here a nice video from Arizona and collaborators published last year. So in pink, you can see a dendrite. And this is, so in transparent here, this is the super resolution morphology of the astrocyte. And in blue to red, calcium activity. And you can see that some calcium signals occur in very point sites near dendritic spines. So there seems to be a link between this calcium activity and synapses. And also Bindochi et al. Pointed that those microdomains occur in defined processes, and they seem not to occur randomly in any location within the cell. They seem to occur at specific locations, but we don't understand yet why is it, what are the mechanisms that are responsible for those microdomains to be observed in specific locations within a single cell, and whether they have functions, for example, the enabling private communication, let's say, between one process and the synapse to which it's contacted. And this is very tricky because as 80% of those calcium signals occur in fine processes that can only be resolved with a super resolution microscopy or electron microscopy, really few is known about their activity. And you can see here the complex morphology from electron microscopy. And clearly we're lacking a lot of understanding of their physiology for now. And what I aim at showing you during this talk is what insights we can gain from spatially extended models regarding those fine processes specifically. So as you know, when we try to modify cell morphology, which is highly diverse in astrocytes and highly complex, when you do this experimentally, you also trigger some physiological changes. And the good thing with modeling is that we can control this, this parameter without changing other parameters, this being able to predict what is the effect of the morphology of the cell or of the processes on calcium activity. It's also very well suited to studying the very small volumes of astrocytic processes, especially if you go for reaction diffusion simulations, and we'll explain a bit more later. And it's also interesting, as we know that calcium channels are not distributed homogeneously in the cell, and those spatially extended models allow to investigate the effect of such various distributions of the different actors of calcium activity on local signals and their spatiotemporal characteristics. So here I will show you one example that we published recently. I mean, not published, but we published it in bioRxiv and it's under review. And for that, we based our modeling study on experimental results from the team of Valentin Negerl in Bordeaux. So the experiments were performed by NISA Arizona. And they have performed STED super resolution microscopy to image the morphology of the astrocytic processes with unprecedented spatial resolution in live tissue. And as you can see, the morphology of the nanoscale is also highly complex reticular morphology. And they noted that there is an alternation between some bulbous structures that are referred to as nodes with thinner structures called shafts here. And interestingly, they have published a paper this year when they applied hypoosmotic conditions to the astrocyte and measured the morphological characteristics of those nanoscale processes. They showed that the swelling of the cell was actually not homogeneous at this nanoscale, and only the shafts showed an increase in size while the nodes stayed the same. And thus we wanted to investigate the effect of such morphology. So this striking alternation between nodes and thinner shafts, as well as its alteration in the diseased brain. This is reminiscent of dendritic spines. And we know with quite some work, both experimentally and computationally, that the width of the spine neck that is connecting the spine head and to the dendrite is a huge parameter that is crucial for dictating calcium activity at the spine, and notably allowing compartmentalization of calcium signals within dendritic spines. And as you can see here on the right panel, you can see that calcium activity that was measured by Arizona et al. In this example, but also in most of the calcium signals that they recorded, they stayed localized in single nodes at the vicinity of spines. So the goal of this study is to investigate whether this link is this fact that calcium signals are in nodes is due to the morphology that we can observe at the nanoscale, and whether the changes of this nanoscale morphology induced by swelling in disease states affect calcium activity. So to do that, we have been using the reaction diffusion model that we have published in 2019 in PLOS Computational Biology. So this is a very simple model that is modeling IP3 receptor mediated calcium activity in single processes. And I won't go into many details. I just wanted to highlight the fact that this model is spatialized and that it takes into account the randomness of reactions in such small volumes. So it means that stochasticity is taken into account. And you can see here the Markov model describing the different states in which IP3 receptors can be. And basically this state here is the only state in which the receptor is open, which is when both calcium and IP3 are bound to activating sites, which lead to the flux of calcium from the endoplasmic reticulum into the cytosol, leading to calcium signal. And first of all, we have investigated the behavior of this model by implementing it with different computational approaches, from mean field non-spatial model to a non-spatial model that does take into account the randomness of the reactions here in black, as well as reaction diffusion models, so both spatial and stochastic here in red. And strikingly, there were no spontaneous openings of the calcium channels in the mean field implementation of the model, which suggested that for this model and in such small volumes, stochasticity is needed for observing spontaneous calcium signals in the model. So then back to our question. So we used this model to investigate the effect of nanoscale morphology of astrocytic processes on local calcium activity. So this is just a reminder of the morphology observed by Arizona and collaborators. And we have used those data and the size of the nodes and shafts measured experimentally to design some idealized geometries that mimic what they observed with STED microscopy. So basically nodes are spheres and shafts are cylinder connected to each other. And to reproduce experimental observations, both in healthy and diseased conditions, we have varied the shaft width, but not the node width in those geometries. And first of all, we wanted to test whether those morphologies were a good approximation of the ones measured experimentally. And to do so, we have replicated some bleaching experiments that were performed by NISA. So basically, there are some fluorescent molecules that are diffusing within the morphologies here in red, Z as green, the same as the experiments. And at bleaching time, part of the fluorescent molecules become non-fluorescent. And then the remaining fluorescent molecules diffuse within the cell, leading to a new steady state. And the time it takes to get to this new steady state of fluorescence level is what we call the time to bleaching recovery. And it's a measurement of the compartmentalization of molecules within the node in which we measure the fluorescence that is here. So you can see here in black experimental traces of bleaching experiments and in red simulations. And you can see here the measurement of the time to recovery of those different traces that show a very similar behavior between experiments and simulations, which showed that those geometries seem to be a good approximation of the morphologies observed experimentally. And it also highlights that when you have thinner shafts, so here if you go to the left side of this graph, you increase the time to recovery, thus you increase compartmentalization. So diffusion flux is lower from one node to another if the shafts are thinner. We then investigated we then investigated the effect of the various shaft width on calcium activity in the nodes. And to do so, we have simulated neuronal stimulation on the first node, while recording calcium activity in both node 1 and 2. And you can see here the calcium traces with different shaft diameters. And what we showed is that as soon as you decrease shaft width, you increase peak amplitude, duration, and probability. And this also means that in case of cell swelling, because there's only the shaft that is increasing in size, this will lead to decreased calcium activity. And we have performed experiments, so those have been performed by Miss Arizona in Bordeaux, in which hypoosmotic conditions were applied to the astrocyte, and we measured calcium activity in processes after those hypoosmotic conditions, which increases shaft width and we observed decreased calcium peak amplitude, which is in accordance with the model's prediction. We further investigated the effect of this nanoscale morphology on the propagation of calcium signals. So to do so, we have performed some successive node stimulations. So at time t0, we were stimulating node 1, and then node 2, node 3, and then node 4, while recording calcium activity in node 5. And we varied one parameter here, which is the time between stimulation of each single node, which is called here tau IP3. And you have here example traces of calcium signals in the 5th node with this delay between each node stimulation that is 250 milliseconds or 3 seconds here. And what we showed is that the time to first peak, so the delay before we measure the first peak in this node number 5 here, increases as you increase the time between successive node stimuli, which is logical. And interestingly, the delay is increased as soon as you increase the shaft width. So despite the fact that when you have thin shafts, you have an increased compartmentalization of signals, actually it increases local activity in single nodes, which then will favor signal propagation, despite the fact that there's this increased compartmentalization, which is a bit counterintuitive and we found quite interesting. And further, we showed as well that when you decrease, when you increase this shaft width, so what you see, for example, in swelling conditions, you decrease peak probability, especially if you have less frequent node stimuli. And we have tested the robustness of this signal propagation within the processes. And to do so, we associated to each node stimulation a probability of stimulation failure. And that you can see here. And basically what you can see is that when the shaft is thin, such as here, even though you skip some of the stimuli, you will still observe a signal in node 5, which means that you can omit some signals and still have signal propagation. So thus you have a more robust signal propagation in those thin shaft geometries, while when the shafts are larger, you can't miss one node stimulation, otherwise you don't see any signal occurring in the node number 5. And we further investigated other parameters in those fine processes that can influence local calcium activity in the fine processes, such as the morphology of the endoplasmic reticulum. And you can see here that if the ER is located only in the nodes, or if it has similar morphology as the processes, you will have completely different calcium activity properties, such as decreased peak duration, amplitude, and probability, which is actually due to a decreased frequency of IP3R opening. And yeah, please note that this is completely arbitrary, as for the moment we have no images of the ER morphology from live tissue at this scale. And finally, we did it a bit actually also in our post-computational biology, so performing some simulations with and without the calcium indicators that are used experimentally to be able to measure those calcium signals. Well, they do alter calcium activity, and we wanted to investigate to what extent the effect of nanoscale morphology could still be observed if measuring free calcium activity rather than calcium G-comp activity. And you can see here some example traces. So on the top, calcium G-comp activity, and on the bottom, free calcium activity. And you can see that the signals are completely different, which is normal because those indicators act as buffers. But interestingly, we could still see a huge increase of peak amplitudes and all other characteristics described so far. I'm not showing everything here, but everything was similar. So increased peak amplitude with when shaft width decreases. So this is our conclusion that we have so far, that the morphology at the nanoscale of the astrocyte processes seems to favor a decreased diffusion flux from nodes, which increases local activity at the vicinity of dendritic spines, and that this allows increased peak probability amplitude, but also signal propagation robustness, and that those features are altered for larger shafts which are observed under hyposmotic conditions, which are lead to cell swelling and are observed in disease conditions such as epilepsy, for example. And this is very important because understanding how signals can be compartmentalized or propagate is critical, as one astrocyte can contact hundreds of thousands of synapses simultaneously. So this is just a video showing to you what we are working on right now. And hopefully there will be a paper on bioarchive really soon. And we are also performing some simulations in more realistic morphologies extracted from electron microscopy. And this helps investigating with a bit more accuracy the effect of cell morphology or molecular distribution on local custom activity. And with that, I would like to thank my team members in Okinawa, as well as my collaborators in France, so Igberi, my PhD supervisor, and my collaborators in Bordeaux, as well as my fundings. And I would like to thank you for your attention. And if you have any questions, I would be glad to answer them.
Thank you very much, Audrey. That was wonderful.
Thank you.
Okay, looks like we have one question from, okay, we have a couple questions. All right, so let's start with Carlos. Let me see if I can find him to unmute him.
Hello, Audrey.
Okay,
beautiful talk. I was wondering this time about what is the delay time between you go from the baseline condition to the swelling condition in biology? How much it takes for the cell to go from one state to the other?
This I must say that I don't remember. It's in the paper from Nisa that was published a few months ago in Glia, if you want to have a look. But really, I don't want to answer because I'm not sure.
So no, it's fine. No worry. But then the question is like, do you think that in the transition between one state and the other, can we find interesting properties, or something can change that is not intuitive, like a linear, let's say, progression from one state to the other?
Yeah, so here for simplicity, I just showed two states, right? But I'm pretty sure that there's continuum between those two, and that probably you want to go directly from one to another. And I have no idea if the changes are linear or if there's a threshold effect. Yeah, this is a good point. This is interesting.
Thanks.
Thank you.
Okay, thank you. And we have another question from Suhita.
Hey, hi, Audrey. Lovely talk, amazing data. So, a couple of technical questions, Audrey. One was, is your IP3 regenerative? And did you hit a bottleneck where if you have enough regeneration of IP3, then you start to see larger or less differences between control and swelling?
So what do you mean by IP3 is regenerative?
So you can so is the IP3 does it just passively diffuse or along the process, or
no?
There are pathways that lead to reproduction of IP3.
Yes, its synthesis depends on calcium by the activity of PLC.
So you do have okay. And so I guess the question
is, if you have a calcium signal, you will have an increase of IP3 production locally as well.
So the question is, did you play around with that parameter to see if the differences get amplified or lowered between control and swelling?
We didn't test that, but that's definitely an interesting question. Also, the effect of the intensity of the neuronal stimulus per se. So actually, I didn't explain this in the talk, but the neuronal activity is very simplified in this model, and it just corresponds to IP3 infusion at the stimulated node. So you can definitely have a more refined implementation modeling of this neuronal stimulus, first of all, and second, you can definitely, we don't know for sure how many IP3 is synthesized in response to neuronal activity, and to what extent it depends on how much glutamate is bound to the receptors on this process. So definitely those are parameters that would be interesting to vary and investigate.
Thanks, sir.
Thank you.
Okay, thank you. So let's move on to the joint discussion, and we can ask more questions there. So I'm going to put, Max, if you can put your video on, and I'm going to also spotlight you. I think you might be already. Okay. All right, great. So yeah, so I don't know if anyone has any questions for the both of them, more general question or for one of them. If not, I do have a question, so I can start with that. Okay, so I have a question that's directed to Audrey actually at this point, and then I have another question for Max if no one else has questions. Okay, so for Audrey, I'm wondering, do you have buffering mechanisms, the calcium buffering mechanisms in your model? So I guess there is some buffering by the GCAM, but do you have any kind of endogenous buffering? And do you think, if not, like, how do you think you would implement that?
So it's,
like
it's a very good point, and we discussed about it a lot with the reviewers of our paper in cross-computational biology, actually. In this implementation of the model, because it's highly computationally intensive, because it's a reaction diffusion in 3D, we have simplified buffering by changing the apparent coefficient of diffusion of calcium. So it's slower and has the same coefficient diffusion coefficient as when bound to the buffers. And we have shown, so I'm not sure, no, it's not in supplemental. So it's in the answer to the reviewers that you can find on PLOS Computational Biology website, as well as in my thesis, actually, I also explained this. We investigated if the model's behavior changed, if we directly included the endogenous buffers in the model or changed the diffusion coefficient, and there was no significant difference at this spatial scale between doing those two. So we chose to keep the simplest because it's less computationally expensive. But definitely, the more buffers you have, the lower the amplitude and the lower the duration and frequency of the peaks. We actually have a figure about this effect also in the cross-computational biology paper. So we're doing this by changing the concentration of GCAM, but actually it's you would see the same for any buffer that you add. And this is actually a very tricky point in the sense that we know which buffers are expressed in astrocytes, but we don't know if their distribution is homogeneous within the cell. We don't know their concentration in astrocytes. We don't know if their expression varies depending on physiological states. For example, if in sweating conditions you have any differences in buffering. So it's super interesting topic, but a lot of data is missing experimentally.
Yes, that's true. There's a lot of limitations still that we need to explore experimentally. So thank you for that answer. But it's nice to know that you did explore that in the model. Okay, perfect. So I don't see any other questions, so I'm going to go ask my question for Max. So if we were to look at more fine-tuned behavior, it's possible, I guess, that the functional connectivity between astrocytes changes over time, or maybe like throughout a trial, a specific behavior. Do you think your methods of exploring this apply to those when this functional connectivity changes, or I guess, what do you see for the future in that?
I think it's a super interesting point, especially I think there was, I mean, there's so many interesting things that are coming out as far as like information content that's represented in astrocyte activity. And I think given that it's very reasonable to think that there is dynamics to this information exchange that's happening inside of the astrocyte network. I think it's pretty tricky using the method. I think there's a big technical challenge using the methodology that I presented with transfer entropy, just because it requires such a huge amount of data in order to actually like come up with a determination for the connectivity coefficients themselves. But that's not to say that that's the only methodology that one could use in order to information exchange between different parts of the astrocyte network. So yeah, I think that's a hugely interesting question. But I don't necessarily have any data that is getting at that dynamics at this point.
Wonderful, thank you.